My name is Luciano Marraffini, and I am the head of the laboratory of bacteriology at Rockefeller University. And my lab basically studies how bacterial pathogens evolve and acquire virulence genes and antibiotic resistant genes. In eukaryotic organisms, basically the driver of evolution is mutation on the DNA. Bacteria, on the other hand, what they do is they share full genes between them, and in that way a bacterial pathogen will pick up a gene that provides antimicrobial resistance from another bacteria. Gene transfer in bacteria happens basically from a donor into a recipient, and genes can transfer in different ways from one cell to another, and if these genes are antibiotic resistant genes, then the recipient that acquires those genes becomes also antibiotic resistant. Bacteria do have pathways that control the sharing of genes, and that's what we study in the lab. And the pathway that we study name is CRISPR immunity. And CRISPR has the ability to block this transfer. In that way, it can prevent the spread of antibiotic resistance. And we're trying to understand in the lab the molecular mechanisms that allow this prevention of gene transfer by CRISPR. What we do daily in the lab is to make genetic manipulations of bacteria and then analyze whether they can become antimicrobial, antibiotic resistant. And the way we do that is we mix bacteria, our mutants, and we provide genes encoding for antibiotic resistance, and then we place them on plates that contain antibiotics. If the transfer is successful, bacteria can grow into these plates. If transfer is unsuccessful, meaning that CRISPR immunity attacked the genes encoding for antibiotic resistance, then the plates are empty. Our aim is primarily to understand how these pathways work, and penultimately, if we understand how they work, we may be able to exploit them to limit somehow the acquisition of virulence genes and antimicrobial resistance.